Okay, welcome back, everybody. It is my great pleasure to introduce a colleague and friend of mine, Professor Jen Shin, from the School of Information Science, or iSchool here at Syracuse University. Her research interests are wide and bridges, I think, the two areas that are most prominent here in the iSchool, the library science side and the information science side, machine learning and AI. Her interests are many and include metadata, knowledge and data modeling, scientific communication, research networks, and research data management. She has received funding from the Institute of Museum and Library Science Services, IMLS, to develop an e-science librarianship curriculum, and from the NSF for the Science Data Literacy Project. Jen directs the Metadata Lab and focuses on big metadata analytics and metadata modeling and linking. Jen Shin will present to us evaluating the impact of collaboration enablers through metadata analytics. So please help me in welcoming Jen. Jen, please.
Okay, thank you. Can I share screen now?
You should be able to share screen.
Yeah. Okay, good. Let me do this. It will take a few seconds. Okay. Thank you, Daniel, for giving me this opportunity to share my research. And from the schedule, I noticed that there are quite a few big names in the science of science field, and which kind of intimidated me. So I'm going to just share some of the results, and okay, I'm not good. And this project is collaborate. I collaborated with my colleague Jeff Hemsley and doctoral student Sarah Brett. Let's take a look at what's in science and science in regard to scientific collaboration, and also the measurements, assessment, and so on and so forth. So I think everyone here, has some background in science of science, research impact assessment. So I'll skip all those essential, fundamental stuff. But we all know in measuring scientific collaboration, two primary categories of measures have been used in previous research. One, we can call it outcome measures, and these are mostly simple count of products over a time period, such as number of publications, number of grants, number of patents, and of course, the authors associated with these products. And the other category is called impact measures. And we all know citation has been used as one of the primary quantitative measures for impact, research impact. And back in 2010, I wrote an article and I figured out that research impact should be measured from three different aspects, because citation is so limited. It measures mostly the intellectual impact, but for others, it's not directly measured. This metric is not directly measured other than intellectual impact. That's the argument for this. So I was reading an article at the time, writing this article. That article talked about how invasive species in the biology domain have made damage, economic and other damages, which created devastating impact for some regions. And I was looking at their equation about how they measure the impact of invasive species. And they talked about the extent. And I think they have another, some other. So I think this equation or this model was inspired by that article. And I thought, when we measure impact, research impact, we should focus on the extent of knowledge diffusion, and of course, citation counts fall into this category, and the rate of adoption of new technology and the discoveries and the benefits to society. All these should be taken into consideration. But of course, we all know, besides the citation, other data are very difficult to collect on a regular routine basis. And extra research has to be done in order to gather those data. And studying scientific collaboration has taken two different routes. One is team science, and the other one is collaboration networks. And of course, these two have some overlaps, but you can see the approaches in these two different domains. Well, I shouldn't say different. In these two related research areas have some overlaps, but the approach, the methods, and the data they use are quite different. So this is just some background information about what I'm going to share for my research. And for the metrics so here I'm focusing primarily on the collaboration networks. The metrics for research collaboration networks have been primarily used co-authorship of publications. And previous study have discovered like small world properties of the collaboration network. And there's also clustering tendencies and power law degree distribution. So these are all well-known characteristics of collaboration networks. However, the data-intensive science brings up new challenges for quantitative science studies. I'm citing this figure from NIH, and in that article, NCBI's article, each year for each GenBank data release, they publish an article to go with the release. So in the one that's published recently, they said from 1982 to the present, the number of bases, that's the DNA sequence or genetic sequence base pairs, the number of bases in GenBank has doubled approximately every 18 months. So if you look at the blue line, and you can see the data growth, the rate of data growth is much faster than the use of the data. Okay, so this, and along the line, you can see all these vertical arrow lines points to when the new tool is created or is developed for using the data. So you can see over time, over the last 30 years, almost, more than 30 years, numerous data repositories, tools have been developed, and which form a cyber infrastructure. I think NSF or the federal research funding agencies like to call this type of infrastructure, cyber infrastructure for research. So given the rate of growth as well as the investment of federal government into the data, research data production and preservation, as well as use. And we feel there are some compelling reasons for including data production in studying the collaboration networks. I listed a few of them here. I think these are the primary reasons, and of course, there are others. Data production is an important stage in a research life cycle. So if we just study the end product publication from a research life cycle, we are missing a lot of information, a lot of stuff along the research life cycle for a research project. Collaboration in research starts before publication of papers, and this is a known fact. Research collaboration data, which here I mean, if we could include the data production data, the data about the data production process, and this can be much richer than publication-centric data in quantitative science studies. And of course, these repositories, data repositories, were established way before, way before the internet. Like GenBank was started in 1984, and the internet started in 1993. So, so the data production has always been an important part of a research life cycle. And the longitudinal collaboration in data production and the publications offers a unique opportunity for us to study science quantitatively. And of course, and we also discover along in our research project that there's a potential for using the metadata in data repositories to track breakthroughs and innovative discoveries. So that said, so we, from 2012, actually, we started doing our pilot test in 2012, and eventually got our first grant from NSF. And later, we continue to, to apply for a grant, and the NSF and the NH were very supportive of our research. So the GenBank metadata analytics project, basically we collect metadata for describing genetic sequences. The genetic sequences including not only DNA, RNA sequences, sometimes they are also protein sequence. I think once a friend who's in biological sciences corrected my terminology. He said we should call it molecular sequences because it covers different types of sequences rather than just genetic or DNA, because they are just part of the whole molecular sequences. So the submission of the sequence data and associated publications and the patents, the time coverage is about 27 years, close to 30 years. And the major network metrics, we run analysis by using, to get the typical statistical properties of networks. And besides, we also develop some new metrics based on data submission, publication, and across examinations. So a typical GenBank annotation record contains the information about the DNA sequence. So if you see the top line, that's the information about the sequence. And then there are authors, the taxon lineage, which is extremely important for the genetic sequence or molecular sequence. And publication, authors, and the PubMed ID, and the data submission information about when it was submitted and by whom, and also other information about the sequence. So in collecting the data, we had a workflow which is completely computational. So the Python code was written to, first download one compressed file. So if you go to GenBank FTP's server, FTP server, then there are very large number of zipped files or compressed files, and some for index, some for DNA sequence. So we have to figure out these and then process one at a time. So download one file, decompress it, and then extract the metadata and ditch the sequence data. And because the sequence data contains very large volume of the file size, and save the metadata records in the buffer space and delete the download file, and then parse the metadata into the MySQL database, and then repeat for the rest of the files. One of the major processing that we did was name disambiguation. To do this, we used the Kaggle solution from the Kaggle solution. I think this is award-winning paper in the Kaggle data, I think it's data analytics or discovery competition. And we also use other sources such as Web of Science, Scopus, and the Microsoft Academic Graph to cross-check the results with other metadata to ensure the accuracy of the name disambiguation. So the nature of GenBank, the GenBank metadata collaboration networks is like this. So for all the authors, some authors will be in the publication network only. Others will be in data submissions only. And then there's also a small portion of the authors who reside in both networks. And so the data is longitudinal in nature, and there's PubMed ID in case we need to check, verify the affiliation information or other information about the publication. And a publication may be referenced in multiple annotation records, and a accession number for each annotation is linked to multiple data repositories, because GenBank is such a critical foundation for many others, like clinical, research, as well as for drug research and etc. So GenBank metadata collection, we have the current version is from 1984 to December 2018, and it is stored in the MySQL database, and the computational analysis runs on our server. And these are some general data. The total number of annotation records runs about 228 million. And the number of publications close to 44 million and 500,000. And then the number of unique authors, a little shy of 1 million. But if you look at the number of authors who are in the publication network only and who are in the submission network only, you will discover the total doesn't add up to the total number of unique authors. That means, these authors have some overlaps in terms of which network they are in. So in, in the in the research process, we develop a new concept. We call it collaboration capacity. and which is defined as the ability of an individual researcher or a team of researchers to collaborate throughout the data production and the publication lifecycle and sustain a network of collaborators over time. So the assumption of this concept is based on a proxy for studying, based on this, the collaboration capacity is a proxy for study scientific capacity. And the multi-stages of a life cycle can be used as a proxy for studying knowledge diffusion and the impact on the level of research productivity and the extent of knowledge diffusion. So this is, our assumption for the collaboration capacity. And to illustrate the concept, we draw this diagram. And on the top, you will see scientific capacity can accelerate or hinder knowledge diffusion. Depends, how effectively the scientific capacity is used. Collaboration capacity is a proxy of scientific capacity. And collaboration capacity can be measured by, for example, data submission network indicators, size of networks, the distribution patterns, core and peripheral positions, temporal features, as well as taxonomic features. And then if we cross-examine these, the data network measures with publication measures, as well as patent data or metadata about patents and the publications. And we can have new measures that were not used before, which include ratio of data submissions to publications, a ratio of data submission to patents, ratio change over time, and the time from data submission to publication, and time from data submission to patents, and properties of collaboration networks in publication, patents, and data submission. So these, bringing the data production data into the study, into the quantitative study of science, offers much more opportunities for us to examine a fuller picture of scientific collaboration. So here are some results from our research. This one is the number of data submissions, publications, and patents. And if you look at this, this point, which is around 1994, about 10 years later, after the GenBank was established, this is the number of data submissions surpassed that of the publications. And if you look at the connectedness, I think this is the giant component. Others remain well connected over time by looking at the giant component size change over time. And also this is the clustering coefficient for data submission as well as the publication. So you can look at, while more clusters of smaller communities emerged. So basically the clustering coefficient has going down, which means the network overall is becoming flatter. And the average ratio of data submissions to publications, before 2000, I think 2003, the trend is very clear, increased very fast. And after 2003, it become leveling off, and so that's the pattern is quite interesting too. And here is the structural shift in collaboration networks. I'm going to play this, which is about a little bit over 1 minute. Okay, so that visualization of networks, and by the way, the network is combined data submission as well as publication, two networks combined together. That's from 1994 to 2018. The reason that we started from 1994 rather than 1984 is because before 1994, the data submission was very sporadic. There were not much activity in data submission. The data submission only took off after 1994. So we collapsed all the data submission data from 1994 into 1994. Okay, before 1994 into that year, single year. So based on the first version, we're still very much focused on how to measure the collaboration capacity. But as our analysis evolved and more results were obtained, we feel that, based on the theory of a SciTech, human capital, which for this, in this theory, and the authors argue that when we measure scientific collaboration, we need to pay attention to how science policy or science funding has enabled collaboration to run more effectively. So I think that is the key part, and which which prompt us to rethink about our model. So in the next version, and we revised our model as the collaboration capacity is something that can be measured by metrics from data production and from the process, data to knowledge. So the sample measures are presented in these two boxes. But I think through looking at collaboration capacity, our goal is to evaluate how effectively the enablers promoted collaboration capacity or helped collaboration capacity is used more effectively by researchers or by teams. So the enablers, for the enablers, we divide it into three categories. One is the SciTech human capital, which means the sum of scientific, technical, and social knowledge, skills, and the resource embodied in a particular individual. And the cyber infrastructure includes data and publication repositories, software tools, and the discovery services supporting data-intensive research. And the science policy, basically ensures resource allocation and the dissemination of research outputs. So this model, you know, and I think for if we think these data production and data technology measures are independent variables, and what collaboration capacity or the dependent variable should be measured. So, and what are the measures for the enablers, and what their relations are. So these are the research questions. We are still exploring them. So conclusion, metadata is science data metadata in science data repositories such as GenBank, Protein Bank, and so on and so forth, offers a great source for quantitative science study. You know, because the new dimensions, it offers us new dimensions to look at teamwork in basic science research at a very large scale, and can be combined with local team data for productivity and impact assessment. And it is rich data, and you know, more analysis need to be done. And so far, we have imported NIH funding data with GenBank metadata, and we have produced a new set. Of course, not all authors will appear in NIH's funding data, but we extracted, you know, match the time coverage, and we have all the research funding data extracted and matched with our GenBank data, and analysis is to be done at this point. We are also collecting policy, technology, and biomedical events for triangulating analysis and interpretation. So this is what we are currently doing and will be doing in the next couple of years. And finally, this research cannot be done without a team effort. This is the current members of the Metadata Lab. We just launched our new website yesterday. So if you are interested to learn more about what research we do, and here's the link. And thank you.
Thank you very much, Jen, for your presentation. I love this data set. I analyze it myself. Some of it, it's a very important topic. We don't do it enough. So we have a question from, or maybe not. Okay, the question disappeared. So again, if you have questions, just raise your hand. I guess I have a question. So I'm kind of surprised that, I mean, data is like so important to us. I mean, in science of science, we can see it very clearly, but I would imagine for the rest of science, it's very important, but it doesn't seem that we are doing this measurement of data itself that much in the rest of science. So it seems like GenBank is doing something right. It's like somehow allowing scientists to submit without, it seems to me that they don't care too much about the publication part. It's important for them. Somehow they are getting credit for sharing data, and it doesn't matter too much that there is no publication attached to it yet. Maybe I'm wrong.
Mm-hmm. Yeah, the growth of GenBank, I think owes a lot of, you know, a big part to the science policy, which is a mandate from federal funding agencies that you must submit your data to GenBank, or you must share your data with a public repository. And this policy also resonated with some major biomedical journals. So for anyone who conduct research, you know, with molecular sequences, they must submit their sequence to GenBank before they can submit their manuscript to a journal for publication. So I think of that mandate, which is, you know, a science policy, motivated scientists to submit the data, and which helped the growth of the data. And I think to think more deeply, I think this mandate also created a culture of data sharing. So if you look cross-disciplinary field, biomedical field is the field that has the best data sharing, data sharing culture compared to other fields. So I think this quantitative study reveals a lot of unanswered questions, and which will likely require qualitative research to find answers. And I think which is what my PhD advisee, Sarah Brett, is doing. She conducted interviews with lab scientists to find out a lot of questions, answers to a lot of questions that we raised during our research, but the quantitative data cannot address.
Yeah, looking forward to hearing those results. Yeah, like this culture, I see it now, AI now, like when they publish a paper, they just put the data set, the code out. But if this is part of the culture in bioscience, that will be interesting to hear from the actual scientists being trained. So looking forward to Sarah's results. Yeah. So we have a question from Stephen.
So I'm kind of following up on Daniel's question. First of all, I think it's great to be looking at these data sets, because I think one of the sort of things that's come up a couple times today is this question of what are outputs besides papers, right? And data is definitely has value. I guess a couple questions. First of all, a comment maybe. Biomedicine may be ahead of other fields, but I feel like many other branches of biomedicine are not doing quite as good a job as GenBank, and I think maybe partly it's because gene sequences are a little bit easier maybe than some other data sets. If you sort of look at the neuroscience world, for instance, there's a strong interest in data sets. But I guess the question is, are there ways of evaluating the quality or effectiveness of data sets? Because I imagine you could just count them as papers, right? And then just kind of treat them as papers and citations and things. But the quality, so often people just like dump their data in a repository without annotating it, and they'll be kind of useless. And so I guess, is there any way of kind of evaluating that and sort of attaching value to the different data sets? And I don't know if you've seen that in GenBank. Can you kind of identify sort of good versus less good data?
That's a great question, because it's critical for if we are going to use this data set for research, right? So GenBank data annotation record, which means it contains information about the sequence as well as associated publications and the sequence data itself, right? So the GenBank is first of all, over time, NIH or National Library of Medicine, and under National Library of Medicine, there's NCBI, National Center for Biotechnology Information. So NCBI is the entity that created all these data discovery services, data repositories, and et cetera. So GenBank is one of the earliest data repository. The NCBI has invested a lot of money and technical expertise to build the tools, and also training the scientists, researchers, how to use the data submission tools. They have at least 2 or 3 different data submission tools. It can be used as a desktop standalone, or now they have the web browser version for submitting the sequence data. So this kind of support for scientists to submit their data to a repository is really comparable for the case of like neuroscience, because it's a relatively small field and it doesn't seem to have that kind of, level of support, money, technical expertise. So, and because this is so important, and also GenBank is the one in the center. So before GenBank, there are two other data repositories. One is called BioProject, and the other one is called a BioSample. And both of these repositories are linked directly to GenBank because DNA sequences can be collected from biomaterials, either human tissue or soil or plants. And so those BioProject, BioSample data, kind of all the result from those projects will be in GenBank. And then GenBank will link to clinical and other curated, humanly curated, manually curated, I think there's a reference sequence repository. So in any case, what all this means is GenBank is relatively well curated, and each submission needs to be approved in order to be in order to show up in the public interface. And they also take batch load, like say from the US PTO, the US Patent and the Trademark Office, take batch load. And so all these, and the batch load, I don't know the quality of those batch load, but so for sequences that have been curated, manually curated, they'll move to the reference sequence repository. And those will be linked to other, more broad repositories. So there's a chain of linkage between the repositories. And so that if anything goes wrong, they have the ability to track to the starting point to verify where there's a problem, what it could be, so they can correct it. So that's the workflow and the relations between these data repositories. So it's a it's a well-designed system or ecosystem of the data repositories, and I think we are relatively confident in terms of using the metadata for research. But of course, there are some other pitfalls we do need to pay attention to, in using such a big data collection for analysis.
So question from Bruce.
Hey, this is really fascinating. I mean, I do think that broadening the set of research objects, artifacts that we track, and we incentivize what we measure, and people respond to incentives. And so the ability to measure and track things here, more broadly is really valuable. I guess I'm trying to understand how GenBank sort of relates to publications. And I mean, is there one or more publications coming out of a particular deposit in GenBank? And then how could I, if I wanted to map something to, say, an underlying health condition or an underlying advance in medical technology, I mean, can you talk through sort of how to sort of, if I wanted to kind of relate it into the broader context and see how it's contributing to health, how is there a way that I can think about how to do that?
Yes, there's a way to track that. For example, for the COVID-19, right? virus, you can search by the taxonomy terms, right? In the NCBI taxonomy, there are standardized terms or class representing this virus, including the variations and et cetera. So you can use the top level term to do a search in GenBank. But of course, the GenBank FTP server is not the place for the average person to do search. But unless you want to download the data collection. But in NCBI, they have other search interface to allow you, or you can even just go to NCBI taxonomy. And once you locate the term, I think the formal term for COVID is SARS-CoV-2-19. That's the official name. So if you go to taxonomy, you can simply click that term. On the right panel, you will see a list of DNA sequences or genetic sequences associated with this. And then you can, so once I did a research, just study the linking mechanisms in NCBI data repositories. So if you know the linking mechanisms where and the taxonomy, the right panel can take you to a predefined search and or to to different data repositories that has information connect to this clinical trials, drug database or patents. And so that by that way, if you want to find a treatment, if you want to identify drug development, or if you want to identify, I don't know, diagnosis or method, and those are the clues that you can follow on to track those information. So that's why I say it's a very well-designed ecosystem that has linking across multiple databases, and to even for lay people, they are not necessarily in the biomedical field, but some family members had this cancer or had this, they can go to that place to search for information to do their own study.
Thank you.
Okay, so with that, I want to thank again Jen for her talk, and we'll jump right